Hello, everybody. This is Shen Zhong from UC San Diego. And our seed network works on human blood vessels. Our lab is interested in discovering RNA's novel functions. I would like to share with you today our ongoing work on natural display of nuclear-encoded RNA on the cell surface. The cell surface is the crucial interface for cell environment and cell-cell interactions. Bioactive molecules, including proteins, glycans, and lipids, are essential for the cell surface functions. However, nuclear genome-encoded RNA, let me call it ngRNA, is not generally expected to appear on the cell surface. Our overriding question is whether the nuclear genome-encoded RNAs, ngRNAs, can be stably associated with the surface of intact cells and exposed to the extracellular milieu. For convenience, I will call these RNAs membrane-associated extracellular RNAs, maxRNA. I will also refer to them as surface RNA. So we are not the first group who suspected RNA being a structural material of the cell surface. In the early 1960s, people observed that cell surface become more negatively charged during metaplastic transformation, where a differentiated cell type transforms into another differentiated cell type. For example, cancer cell surface is more negatively charged than untransformed cells. Such increased negative surface charge was commonly attributed to increased glycolysis and presentation of negatively charged glycans on cancer cells. Nevertheless, a pioneer biologist, Leonard Weiss, asked, is there any other material that might be responsible? So Leonard Weiss incubated leukemia cells with radioactive uridine and found RNase-sensitive radioactive material at the cell periphery. So Weiss wrote, these results are interpreted to indicate that RNA is a structural component of the peripheries of these cells. So Weiss called them periphery RNA and found such periphery RNA molecules modulate cell surface charge. A few years later, Canadian biologist Sarah Zelick also reported RNA-sensitive materials on the cell surface. So Sarah Zelick was aware of Weiss's work. She cited Leonard Weiss's papers, but did not conclude that such materials must be RNA. She wrote, the nature of this material is currently under investigation. So these papers seem to have gone nearly ignored for half a century. Our goals and approaches are as follows. So we will start with a lymphoma cell line to test whether there is RNA on the cell surface. To do such a test, we first developed a technique to sequence maxRNA to identify candidate maxRNA genes. Then we will use single molecule RNA fish to validate any candidate maxRNA. Second, we will test if primary and normal human cells present maxRNA. If so, is there a cell type specificity of the maxRNA presenting cells? Third, we will identify what maxRNAs are presented on human primary cells. Finally, we will test if a specific maxRNA can mediate cell-cell interactions. Furthermore, we will test if a specific region of a maxRNA can mediate cell-cell interactions. We developed a technique called Surface-seq to sequence maxRNAs. The basis of Surface-seq is a nanotechnology from my colleague Liangfang Zhang's lab. Liangfang's lab can extract plasma membrane from cells and tightly assemble this membrane around a polymeric core to form membrane-coated nanoparticles, MCNP. This technology suits our purpose because it retains the inside-outside orientation of the membrane by keeping the surface molecules of the membrane facing outwards. Leveraging Liangfang's technology, we developed two variations of the Surface-seq technology. In variation A, after MCMPs were assembled and washed, RNA was extracted and subsequently sequenced. In variation B, shown at the bottom, after MCMP assembly, the RNAs exposed on the outer surface of the coated nanoparticles, which correspond to the outer cell surface, were directly ligated to 3' RNA adapters. The RNA was subsequently purified and ligated with 5' adapters. Because the 3' adapters were selectively ligated to the outside-facing RNA, this technical variation enriches for the outside-facing membrane-associated RNA in the sequencing library. So we generated 5 Surface-seq libraries from a lymphoma cell line. So each sequencing library reviewed approximately 200 link RNAs, and 82 of such link RNAs, including MALAT1, were shared among the 5 Surface-seq libraries. To validate these candidate maxRNAs, we carried out single molecule RNA fish on the cell surface without permeabilizing the cell membrane. In the test of MALAT1 as a candidate maxRNA, nearly all cells treated with MALAT1 antisense probes exhibited surface fish signals, whereas most cells treated with mismatch control probes exhibited no signal. To confirm that surface fish signals are not a result of RNA leakage from damaged cell membrane, by a transmission through dye microscopy analysis, where only live cells with intact membranes are fluorescently labeled, as shown in the lower right panel. So together, this data suggests that the presence of specific nuclear-encoded transcripts on the surface of intact live cells. So we used human peripheral blood mononuclear cells, PBMCs, to test if any cell type is more likely to present maxRNA than other cell types. We probed for putative maxRNAs on PBMCs by hybridization with a randomized library of fluorescent labeled oligonucleotides. Hereafter, I will refer to this technique as in situ surface fish, IS fish. We co-labeled human PBMCs with cell type specific surface protein markers. So both imaging analysis and FACS analysis suggested that CD14 plus PBMCs are more likely to also present maxRNA than other cell populations. To further test if maxRNA is uniformly presented in all cell types, we subjected the maxRNA presenting and maxRNA negative cells to single cell RNA sequencing. The single cells expressing monocyte markers, CD14 and LYZ, were enriched with the cluster of maxRNA presenting cells in the circle. This data corroborated the idea that monocytes as a major type of maxRNA presenting cells. So we pulled down maxRNA and subjected them to sequencing to obtain specific maxRNAs for further functional validation. To account for different types of potential artifacts, we developed 3 technical variations and generated 6 control sequencing libraries. So we identified maxRNA from each technical variation as well as their overlaps, shown in the Venn diagram. Each Venn diagram is a technical variation. So we choose 10 maxRNA candidates for functional tests. We choose the monocyte endothelial cell interaction assay as our testbed assay for cell-cell interactions. So we blocked each specific maxRNA by extracellular incubation of the antisense oligos of each RNA with monocytes for a short amount of time. After the blocking step, we seeded the monocytes onto confluent human endothelial cells and measured the relative proportion of attached monocytes. The monocytes incubated with antisense probes against 2 maxRNAs, namely the FND-C3b, the CMT-M6, exhibited reduced monocyte attachment levels. This data suggests that blocking a specific maxRNA can attenuate monocyte endothelial interaction. to test if any specific region of a maxRNA is responsible for the reduced monocyte-EC interaction. We repeated the above experiments with singular antisense oligos. So based on Surface-seq read coverage, so we did IS-fish and sequence them. We choose 9 oligos against different regions of the FND C3b messenger RNA. So these different singular antisense oligos were plotted at the bottom from E1, E2, E3, E4, U1, U2, U3. So these singular probes cover, some of them were targeting different exons, and from U1 to U5, they were targeting different parts of the 3' UTR. We observed nearly no effect from 5 out of 9, the 9 blocking oligos, namely E1, E2, E3, E4, and U1. However, each of the remaining 4 oligos reduced the monocyte attachment levels. These 4 oligos marked there as U2, U3, U4, U5 are antisense to the 3' end of the 3' UTR. Consistently, so there are reproducible maxRNA sequencing peaks at the 3' portion of the 3' UTR. So this data suggests that not all parts of a maxRNA gene would equally be important for their cell surface functions. So in summary, our data substantiated Nearnet Weiss's theory of RNA being a structural component of the cell membrane. We found maxRNA presenting PBMCs to be enriched in monocytes. Blocking specific maxRNA on monocytes can interfere with monocyte endothelial cell interactions. So the heroes and heroines of this project include Norman Huang, Xiaochen Fan, Cassia Zeleta Rivera, Xu Nguyen, and Zhangming Yan from our lab. And in Liangfang Zhang's lab, especially Rongli Fang, is indispensable to this work by bringing in their nanotechnology. Zhen Chen's lab, especially Yinjun Luo, are critical to this work by bringing in their endothelial cell expertise. Thank you, everyone. I'm happy to take questions.
Thank you very much, Zhang. Very interesting. Any questions for Zhang on maxRNA? I saw Pascal and Lars both raise their hands, so.
Yeah,
Pascal, go ahead.
Thank you for this very fascinating topic and presentation. What happens with regard to the TLR signaling? Have you checked that toll-like receptors are not interfering with this RNA?
Absolutely. So T-cell toll-like receptors are known to interact with viral RNA as a way, perhaps stabilizing alien RNA with cell surface. Whereas here, our primary interest was to ask whether any human genome-encoded RNA could have been presented at the cell surface. So toll-like receptors still are one of our primary targets for protein validation. We try to pull down potential cell surface RNA and carry out mass spec on the cold pull down proteins. I didn't have time, and we didn't really have thoroughly analyze the data. It didn't look like that toll-like receptors would be the primary. It could be other transmembrane proteins could have served a role for this human genome-encoded RNA, at least in the PBMC that we try to put down. That said, we haven't really stimulated the PBMCs with different viral pathogens. If we were to do that, we could anticipate to see a difference.
Mm-hmm. Okay, thank you.
Lars, do you want to go next? And then we'll go to Gary and then probably move on.
Yes, very nice talk and obviously many implications for such a finding. So I was wondering, are these cells are these live cells, and how long do you treat them with the blocking oligos? Is there any chance that these oligos get taken up by the cell and act as kind of antisense probes, impacting any of the gene expression inside the cell and having an indirect effect on attachment?
Indeed, Lars, that's an important question. That is also part of our intense tests. The question is there any chance that these antisense oligos could have taken into the cells and serve more like an RNA mechanism rather than extracellular mechanism? So it turned out that for monocyte EC interaction, this assay, since its invention in the 1990s, people have used it a lot in combination with RNAi. Because the cellular phenotype somehow is quite quantifiable. So there have been a large amount of literature in characterizing how to optimize RNAi protocols and using a very similar monocyte EC response. Turned out that the best that people have seen any effects, at least in the published domain, were overnight, not only incubation, but with either lipofectamine or other ways of transfection. It would require a transfection agent with overnight incubation to see any quantifiable cell-cell interaction response up to the best in the literature. Here, on the one hand, we didn't have any transfection reagent in there. The total incubation time was less than 1 hour, less than 60 minutes sharp. And we also quantified the corresponding protein from the monocytes and carefully, many times, quantitatively. There was no, didn't expect any change, and there's no way to see any change for the corresponding protein.
Excellent, great.
Gary, do you have a quick question?
Yeah, we're interested. Great talk. We're interested in cell-cell communication networks, and I'm just wondering if you understand how much selective information there is between cells, or is this more of a structural cell-cell connection, or is it also maybe involved in cell communication?
Indeed, those are the two major functional consequences that we are trying to tease out. Here, all that I can offer are conjectures. On one hand, apparently there's a structural implication, there's a charge implication, but on the other hand, RNA is information rich. In 6 bases, it's a huge coded information in there. So this could lead us more thinking toward that this may not be just a structural component, although it very well can be, but there's so much information in there now exposed on the cell surface as a information-rich flag for the cell.